Hi, thanks for having me here today. I'm very honored to have the opportunity to present this work to y'all. I'm going to admit that I crammed a whole lot of information into the first half of this talk, so I'm going to fly through a lot of history of inflammation and perinatal brain damage. It may make you dizzy, but I will do my best to bring it all back around at the end, and it will become clear. So in terms of inflammation in the relation with perinatal brain damage and infection in perinatal brain damage, we know that microbial invasion of the amniotic cavity can contribute to perinatal brain damage. We have evidence to support that this might be the case in a number of situations. Now, if you were at last year's keynote presentation with Dr. Roberto Romero, you've likely seen this figure here that I'm about to show you. Now, the idea of the microbial invasion being a potentially direct insult is happening with, there's the presence of the pathological organisms in the vagina or cervix. These gain access to the intrauterine cavity, and they reside in the decidua. And the bacteria can gain access to the fetus by aspiration of infected fluid or by direct invasion, seeding into fetal circulation, etc. And this can actually attack the brain. Now, when we look at the Elgan study, we see that the microorganisms, they don't have to exactly elicit an inflammatory response in the placenta or in the umbilical cord to be associated with elevated risk of cerebral palsy. So the idea being that microbial invasion without the host response, the host inflammatory response, can actually be associated with elevated risk and might contribute to that and be the cause of brain damage in some cases of CP. Now, we also have evidence that intrauterine infection, meaning that there's a maternal and or fetal inflammatory response to that microbial invasion of the amniotic cavity, can contribute to perinatal brain damage. Now, this has been thought of for a long time, but later studies have looked at the measurement of inflammation-related proteins in the amniotic fluid and showing that the higher concentrations of interleukin-6, the higher the risk of white matter injury and cerebral palsy. Now, also looking at the fetus and the fetal inflammatory response reflected into histologic inflammation of the umbilical cord, and also looking at the cord blood IL-6 concentrations. Higher cord blood IL-6, higher risk of neonatal morbidities and evidence of brain injury. Now, there's also the idea that non-infectious stimuli can also initiate maternal and or fetal inflammatory responses. Now, this idea has actually been around for some time. In the 70s, investigators performing autopsy studies had proposed that timing and severity of infection may explain the variability in brain damage, and also that non-infectious inflammatory stimuli can contribute to perinatal brain damage. So this concept has been around for some time. Now, an example of one of those might be maternal antifetal rejection as a potential non-infectious or presumably non-infectious antecedent that could lead to perinatal brain injury. And we also know that failure of inflammation resolution processes, this can also contribute to perinatal brain injury. Now, this has also been around for some time as well. Following some rubella and CMV epidemics, it was noted in the late 1960s that besides the teratogenic effects of the virus, a chronic disseminated infection often ensued, and this caused, among other things, encephalitis, brain injury. Now, we know that in adults, chronic inflammation and the failure of inflammation resolution processes is associated with a wide range of multi-system organ damage. So this isn't a good thing to have. We have some very preliminary evidence that chronic inflammation in the placenta, so chronic chorioamnionitis, might actually prime the fetal brain or cause some circumstances that makes you more susceptible to subsequent inflammatory events. So what I'm showing you here is that for newborns that have been exposed to maternal chronic chorioamnionitis, so they have chronic inflammation in the placenta identified by histologic examination. If you have both that chronic chorioamnionitis and then you also have the fetal inflammatory response as evidenced by phonocytis, that you have a higher risk of neonatal white matter injury. However, if you have only one of these two exposures, you do not have an elevated risk of neonatal white matter injury in this one study. And this is a single center study, very preliminary, but interesting nonetheless. Now, there's traditional and also recently proposed models of how the fetal inflammatory response contributes to brain damage. The traditional view was that you had a trigger, it hits the innate immune response, and then this is superimposed upon well-formed structure. and then that damages the structure and then results in damaged function that you would see clinically. Now, the proposed view that is emerging is that you have a trigger, and then there's something that happens with a feedback loop. There's some bounce back between the innate and the adaptive immune system. And during this continuing process, this bounce back between the innate and adaptive immune systems is superimposed on developing structure rather than developed structure. So the timing during gestation, the timing during development is critical, and whatever is triggering the innate and adaptive immune systems, together there is some sustained process that appears to be contributing to the damage, not necessarily some linear or direct relationships. Animal models support this view. We can see here we're looking at the phases of inflammation responses to acute stimulus. And the point here is that the phases can last, there's an acute that can last minutes, a secondary phase from hours to days, and then a tertiary phase that seems to last weeks, if not years. Now, expressed differently, this is the number of inflammation-related proteins and the timing of this inflammatory response in phases to an insult. And again, this lasts up to weeks, maybe even years, in terms of particularly the resolution of chronic inflammation. Now, looking at intermittent or sustained systemic inflammation is a modern, a very important concept in terms of what we think about cerebral palsy and multiple forms of perinatal brain damage in the extremely preterm newborn. The idea here is that you can have a stimulus that is either infectious or non-infectious. And this causes some type of an inflammatory response and then can cause, again, this feedback loop in terms of something happens to sustain it over time. Now, this might be continuous or it could be intermittent. It could be a sputtering effect. And the things that might be involved in causing this type of a sustained phenomenon is you have persistence of inflammatory stimuli, so you have something that keeps stimulating the response, impaired inflammation resolution, developmental regulation. Again, there's feedback loops between the innate and adaptive immune systems, epigenetic mechanisms, and other possibilities. Now, animal studies support the idea that persistent inflammation is a problem. 35 days after a local, apparently non-inflammatory set of brain-damaging stimuli, immature rats, microglia, macrophages, astroglia, and CD4 lymphocytes in the brain remain activated. So the point here is 35 days later. Now, when you have lipopolysaccharide stimulus to the pregnant rats, it's followed by apoptosis of brain cells in their newborn starting a week after birth. So you have the stimulus during pregnancy, but you see what's going on with the pups after birth, a week later. Now, also 3 months following a hypoxic ischemic insult to the brain of mouse pups, activated T lymphocytes are still present in the damaged hemisphere, again, 3 months later. So the idea that animal models are supporting this importance of sustained inflammation. Now, looking at human studies, also we have evidence of sustained inflammation being important. School-age children with CP, they have higher tumor necrosis factor plasma levels than controls, and their monocytes exhibit a more vigorous TNF response to lipopolysaccharide stimulation than monocytes from controls. And again, this is at school age, so this is long after birth. Now, the presence of astrocyte damage marker S100B in the systemic circulation of children with CP also suggests that disease processes might be ongoing or sustained. Now, the information about this that really supports this, that is most impressive, comes from the Elegant study. This is a multi-center study with around 1,500 newborns delivered before 28 weeks across 14 institutions. And among the investigators is our own Nigel Paneth. Very impressive study here. Now, the persistence after birth is systemic inflammation, defined here as an inflammation-related protein in the highest quartile for the cohort for gestational age at delivery. Okay? Now, this is associated at birth with umbilical cord inflammation. But interestingly, the umbilical cord inflammation is associated with persistent inflammation on day 7, but not on day 14. So there's something that's not necessarily a linear consequence of the fetal inflammatory response resulting in something in the newborn, and then you can see it persist all the way through. It's not as clear as that. Now, after birth, persistence of inflammation is associated with white matter injury. You can see in terms of the ultrasound and ventriculomegaly or echoleucent lesions. At age 2 is associated with higher risk of microcephaly, severe developmental delay, and also cerebral palsy. But are there modifiers of the association between what is referred to as ISSI, or intermittent or sustained systemic inflammation, and perinatal brain damage? Here is a study that we did last year, and we were focusing on hyperthyrotroponemia, a fancy word for having TSH concentrations in the highest quartile for gestational age in the cohort, and looking at whether or not there's a difference in risk of multiple indicators of brain damage, including cerebral palsy, by the presence or absence of systemic inflammation. Now what we're going to look at here, we're about to go through 600 odds ratios. So this, we're going to be, I'm going to explain this figure, and then we're going to go through it somewhat slowly. In the vertical axis, you've got about 28 inflammation-related proteins. Each one of these rows represents one logistic regression model, and each of the different inflammation-related proteins is used to define intermittent or sustained systemic inflammation. Now the horizontal axis is the odds ratio, and the first column is the study group. And this study group is simply, these are kids who had a high TSH quartile concentration for gestational age, but they did not have the inflammation-related protein in the highest quartile on 2 separate days, 7 days apart within the first 2 weeks. That is ISSI. Okay? So high TSH inflammation-related protein is not in the highest quartile. The second column is the opposite. It is you have the high ISSI, so you have a highest quartile inflammation-related protein, but your TSH is not in the highest quartile. This third column is you have both. So you have both the high TSH and you have the highest quartile inflammation-related protein for gestational age. Now, the lower part of this figure, what you'll see repeating over, is where I tell you what the outcome is. So right here, this is quadriparesis. Now, looking at HTT alone, you can see elevated risk of quadriplegia, around 2-fold elevated risk across numerous definitions of ISSI. Now, when you look at the systemic inflammation, not so much. Only one model out of the 28 showed an elevated risk. When you put these together, you really don't see any combined effect. However, if you were to look without stratifying by the presence of the systemic inflammation, there is no association between high TSH concentrations and quadriparesis. This association only becomes apparent when stratifying by the presence of systemic inflammation. Now we're going to look at another outcome. We're looking at diparesis. So again, we have a different pattern of association. Highest TSH quartile, there's really no association at all. A little bit, only one model with systemic inflammation shows elevated risk. And when you put the two together, you see something not much going on there. Now when we look at hemiparesis, you get a different pattern. So when you have just the high TSH concentrations, nothing's going on. But yet you have systemic inflammation, several models show elevated risk, up to a 5-fold increase in risk, and nothing with the combined effect. And I promise you this, the combined effect will come in here in a second. So ventriculomegaly, you're seeing nothing with TSH, a bit with systemic inflammation, but now all of a sudden you're starting to see a combined effect. So if you look at the bottom half of the figure, you can see a 1 odds ratio for high TSH, no association of 1 odds ratio for systemic inflammation, but you put the two together and in a handful of models, you have all of a sudden about a 2-fold increase in risk. And then if you look at some with the systemic inflammation alone, when you add the high TSH concentrations to that, it exacerbates that risk and makes it even higher. Now, this is an interesting one that I really can't explain, but I find it interesting. This is the hypoechoic lesion. If you have just the high TSH concentration in numerous models, you have a highly protective effect. So you have a much lower risk of an echolucent lesion if you have high TSH concentrations, but you do not have the inflammation-related protein in the highest quartile 2 separate days apart. You really don't see much of an association with systemic inflammation alone, nor with the two apart, or two together. Now, when you look at very low mental developmental index, no association with TSH, some with systemic inflammation, but again, much more models become apparent when you add both together, and the overall absolute measure of risk becomes higher when you have the combined effect, suggesting modification. Very low psychomotor developmental index, you see the same pattern. A couple of signals of, less than a 2-fold increase in risk, so nothing too substantial there. Not much of systemic inflammation alone, but a handful of models, you put those together and you're between a 2- and 3-fold elevated risk. Now we look at head circumference, less than the z-score below n of 2. Nothing with TSH, a little bit with systemic inflammation, but again, when you put the two together, you have a higher pattern of risk and a larger number of models are showing the elevation, again suggesting effect modification. Now this is the overall pattern. The red means that you're associated where both TSH and systemic inflammation is associated with higher risk. Yellow means only TSH is associated with higher risk. Orange is only systemic inflammation, and then green is protective. So you can see that the pattern of risk differs across the complications and indicators of brain damage, including cerebral palsy, depending on this combined effect. Now, interestingly, animal models suggest the same thing. And after we published our study, there was an addendum to a commentary in Nature Neuroscience talking about thyroid hormone trials and saying that what we were seeing was what some have seen in animal models, and saying that the failure of some studies in animal models of showing an impact of thyroid hormone on the prevention of brain injury potentially could be explained by the failure to stratify in systemic inflammation. Now again, remember the highly protective association we saw with echolucent lesions was only apparent when we said you did not also have systemic inflammation. And the animal models support that this might be the case, and this might have implications for the design of therapeutic trials. Now, the next one, the associations get more robust and be prettier colors to look at. We looked at elevated endogenous erythropoietin concentrations and their association with brain damage. Now, looking at just the EPO concentrations in the highest quartile, you see an association with very low mental development index and very low psychomotor development index, less than 55, and also microcephaly assessed at age 2, about a 2-fold increase in risk. So this is if you're looking with just the EPO concentrations. Now, again, looking at the same figure that we were looking at before, the first column is you have a high EPO, highest quartile concentration, but no inflammation-related protein in the highest quartile. The second column is you have the systemic inflammation, but you do not have EPO concentration in the highest quartile. The third column is you have both. Now, this is for quadriparesis, and you're looking at no association if you look at EPO without the systemic inflammation. Systemic inflammation, you see a little bit of risk with only 2 models, a protective effect that we can discuss maybe at another time with IL-6R. That's quite interesting. And nothing here looking at the combined effect for this outcome. But when you look at diparesis, no association for EPO alone, a couple models with the systemic inflammation, nothing with the combined. Hemiparesis, similar pattern here, but all of a sudden when you look at the combination, you have a number of models that are showing you a significant elevation in risk. And this elevation in risk is up to the magnitude of a 5-fold increase in odds. So to go from an odds ratio with either one exposure or the other of 1 to increase to an odds ratio of 5-fold greater risk with the combined effect is rather impressive. Now for ventriculomegaly, you see nothing with EPO alone. You see more than several models with systemic inflammation. But again, when you look at the pattern of risk, when you see more models with systemic inflammation and a bit of higher risk going on here. Now the hypoechoic lesions, nothing with EPO alone, systemic inflammation a little bit and not much going on here. But now we're getting into the robust patterns of association with a very low mental development index. Almost every model shows that if you have high EPO concentrations, you're at a significantly elevated risk of having very low mental development index. Systemic inflammation by itself is not as associated, but you put the two together and all of a sudden the systemic inflammation is very important, and the odds ratios are bordering around 5-fold in a number of models. You see the same pattern of association for very low psychomotor developmental index, where hyper-EPO is associated, but you put it together with systemic inflammation, it gets worse. Whereas if you have systemic inflammation, but you do not have the high EPO concentrations, you really do not have that increased odds of having very low psychomotor developmental index scores. Now with head circumference, you see a little bit of a signal with EPO. You see much more of a signal with the systemic inflammation, but again, the odds ratios, you see a little bit more in terms of the combined effect. Now here again is the same figure we show, that's like a heat map to show you how the patterns differ across the complications. And you can see that for the very low mental and the psychomotor developmental index scores below 55, EPO alone is definitely associated, but when you put it together with systemic inflammation, it's a much more robust pattern of risk. And you can see that for some, EPO alone, for instance, with the hemiplegia, EPO alone only in a couple models was associated with higher risk, but if you put it together with systemic inflammation, it's a much higher risk. Now, we don't know if this is causal or in any way. This could just be that the kids who have the EPO concentrations in the highest quartile had a more severe insult. This could be that this is part of the inflammation resolution process. It could be that they had a very rough injury and that this is the body's inflammatory response to try to resolve that damage. So this is not to say that this is a causal association. This is to say that we're looking at this from a systems perspective to try to figure out the balance between what's going on with the original systemic inflammation and what can we be looking at to see whether or not it's something with the resolution process of that inflammation. Now, there are some trials going on to look at EPO in terms of whether or not you can prevent brain damage, and we hope those will work, and we're hopeful that that will be the case. We don't know how our findings here of looking at endogenous concentrations, how they are associated with anything that has relevance for these trials, but maybe so. So some conclusions, because there could be a lot, because I just flew through a bunch of information for you. I think we went through over 800 odds ratios there. From a systems perspective, the first conclusion is the relation between an indicator consistent with the paucity of protective factors. So if I look at the high TSH concentrations and I consider, well, I might not think that high TSH is going to be a problem, but I might consider that those kids with high TSH concentrations, they may have delivered earlier, they may have something maturationally different, there may be something reflective that we can't measure and can't see at this time. So it could be reflecting a pathway of the paucity of protective substances. Looking at it in that way, it's interesting to think that the relation between that and brain injury depends on the presence of intermittent or sustained systemic inflammation. And as I noted, based on the animal studies, this may have relevance for the design of trials and maybe even our own thyroid trial with Nigel here. We'll see. High EPO concentrations on postnatal day 14 seem to modify the relationship between systemic inflammation and indicators of brain damage. Hyper-EPO, regardless of systemic inflammation, is associated with elevated risks of very low MDI and PDI and microcephaly, but not with any of the cerebral palsy subtypes. However, when you have both high EPO and you have the systemic inflammation, you're at higher risk than others of very low MDI and PDI, ventriculomegaly, hemiplegia, and microcephaly. So those things become apparent only when you stratify based in that way. Now, the interruption of processes contributing to maternal and fetal inflammatory responses, these might result in prevention of perinatal brain damage. As we talked about everything from chronic inflammation, acute fetal inflammation, and there's also neonatal inflammation-initiating illnesses that we had looked at in a multi-hit model, saying that the more inflammatory conditions or insults that you have, the worse off the brain is. Now, this means that there might be multiple windows of opportunity to interrupt these pathways and to prevent CP. And animal models support this view, and these may happen during gestation, these may happen at birth, these may happen after birth, up to months, if not longer after. We don't know, but the evidence now is showing that there could be a plethora of possibilities for this. So it's a very exciting time to look at these things, and I'd like to say that, thanks for the financial support, and for my acknowledgments, thankful to Alan Levington, Liz Allred, and the Elegant study investigators, including Nigel. The authors acknowledge the contributions of their subjects and their subjects' families, as well as those of their colleagues, and all of the participating institutions, including that in Lansing with Nick Alumu here. So I'd like to say thank you for your time and attention. I very much appreciate it. Questions?
A statistically naive question. Some of these outcomes are continuous variables. I'm curious why when you look at this goal, that kind of asking.
Well, for the MDI and the PDI, so that would be the mental developmental index and the psychomotor developmental index, it really follows the system of thought that we, in terms of IQ, and being that those that are very, very low, so the less than 55 that are extremely rare in the population are more likely to reflect biological antecedents, whereas those that are in the moderately low range may reflect a more complicated pattern of risk. So if we're looking for biological phenomenon, trying to understand the underlying pathway, we look at the very low and then concentrate less on that moderate way. I did model those. It is associated, the same pattern. I just figured that the near 1,000 odds ratios was enough, but it's a similar. But that was the school of thought in terms of doing those types of classifications. Pete?
Steve, do you know if the ongoing EPO trials will allow for a post hoc division on inflammation status?
I don't know, because this idea, the Balava lab and then Pierre Grisson's, they're showing in two separate ways with two separate models that systemic inflammation, there's something important, and that has relevance in terms of looking at these. But this is a newer idea, and so I don't think it has gained traction. I think that we're helping with that situation by showing that we see something loosely analogous in what we can do with observational research. But the idea here is, from the system's perspective, to take the animal research, merge it with what we can do observational to design better trials sooner so that we can get to a preventive strategy that works to alleviate suffering sooner than we would do working in silos. So that's kind of what we're going for. But whether or not this has actually been applied in terms of stratifying on that, I'm not aware of that at this time. Oh, right.
The idea that inflammation can be sustained, perinatal inflammation for a few days or weeks after birth, I think is very solid. The idea that it really persists into childhood, I saw the same small papers, there were small samples, and I don't know about the controls. How strongly would you weight the evidence in favor of really the inflammation being sustained in children with CD or children, not just babies?
The answer is I don't know, and what I would make a guess if forced to, I would say probably not much, but showing the concept that even that far after the point of birth, that you pick up a signal, whether it's causal, whether it's anything, it intrigues the imagination, and it allows you to look earlier. And if you look at sustained inflammation at school age, and then you start to look backwards, the further back you look, the more important the sustained inflammation becomes. So is it happening at school age? I don't know. But I would bet money that it is happening in the month after birth.
Steve, I just want to comment that there's an investigator from Pittsburgh named Amy Wagner with traumatic brain injury, and she has found increased inflammatory states 17 years after the initial injury.
If you look at Pierre Grisson's tertiary damage paper, the review, it's a fabulous, fabulous review on tertiary mechanisms of perinatal brain damage. If you read that, the possibilities of it lasting that far become plausible to a far greater degree. But in humans, I don't know, because we haven't had studies. I mean, we have a hard enough time getting studies funded to follow people for 2 years, let alone for longer than that. So I'm not so certain about that, but I'm excited about the earlier part. Hopefully, if we can intervene earlier, we won't have to think about that, about whether or not that works later.
Let's take one more question.
So if inflammation lasts for a long time, are there treatments? What do you do?
Exactly. We want to find out. That's the exact point, is that we could possibly intervene in gestation at the point of birth, shortly after birth, and maybe even 12 years later. It remains to be seen, but it's plausible. It's a plausible pathway. It might happen.
Are there obvious candidates for a medication that would do that sort of?
There's a number of candidates that are out there. That could be a very exciting talk about dendromeres, nanotechnology, all sorts of different things that are looking to be extremely promising. It's an exciting time to be in science, and I expect that in the future these will become successful.
Dr. Kaufman.
I'll catch you back there. Was there any comparison done between the mother's numbers and the infants to see if she had perhaps impacted that at all? Is there any studying about maternal history with that?
Yes, the pattern of systemic inflammation in the newborn differs by the pregnancy complication of the mother. Now, do I think that's a direct association? I don't think so, because the pattern of the clinical diagnosis of the mom is really reflecting the gestational age at delivery. And the earlier the gestational age at delivery, the higher the inflammatory cascade. So I think that could be a proxy indicator, but I don't know with certainty at the time, but it's possible.
Thank you.
All right, thank you so much, everybody.